First, we need to obtain a DNA sample. This can be done by collecting cells, blood samples, or swabbing the surface. This sample might contain a mixture of cells, so the DNA has to be isolated from the other cellular components before we begin PCR. To break open the cells and release the DNA, we use something called a lysis buffer. The lysis buffer contains chemicals that weaken the cell wall or membrane, allowing the DNA to spill into the solution.
Next, we incubate the sample in a 99 degrees Celsius water bath for about 5 minutes. This high temperature helps to further weaken the cell wall, and it can also break down any proteins that might be contaminating the DNA.
Now that we have our DNA sample, it's time for PCR.
But what is PCR and why do we need it?
What is it? Glad you asked. PCR is a technique that allows us to make millions of copies of a specific DNA sequence. This amplification is crucial when we have only a small amount of DNA, but need a large quantity for future such as bacterial contaminants.
Tell me 3, 2, 1 or something. Shit.
Go!
Nucleic acid, the building blocks of DNA.
Nucleic acids provide the raw material needed for DNA synthesis. Short pieces of single-strand DNA that prime the DNA for replication by providing a starting point for the Taq polymerase. These are known as primers.
Taq polymerase is a heat-resistant enzyme that adds nucleotides to the DNA strand, building the complementary DNA sequence. Template strand is the DNA that contains the sequence we want to amplify. The template provides the information needed for making new DNA strands.
First, we heat the sample to 94 degrees Celsius for 30 seconds. This step, called denature, causes the double-stranded DNA to melt and separate into 2 single strands.
Okay.
Next, we cool the sample to 54 degrees Celsius for 30 seconds. During this annealing phase, the primers bind to their complementary sequences on the DNA template, providing a starting point for the next step. Finally, the temperature is raised to 72 degrees Celsius and Taq polymerase begins to work. It's extending the DNA strand by adding nucleotides that match the template strand. This step takes about 60 seconds.
After 1 cycle, we've made 2 copies of the DNA. 2 cycles, 4 copies. The number is exponentially, meaning the more cycles we do, the more DNA we get. So if we do cycles, we'll have amplified to our target DNA to millions of copies. It says insert your age and birth month here. Oh, son of a...
Me? No. I don't know tanning and like. I usually go like
Literally.
Stop. Okay, go.
Okay, once we've amplified our DNA with PCR, we need to analyze it. This is where gel electrophoresis comes in. The purpose of gel electrophoresis is to separate the DNA fragments by size, so we can visualize and measure them.
How do we use gel electrophoresis and PCR to distinguish bacterial contaminants?
Word.
Okay.
So you can't have
Just not. By combining PCR with gel electrophoresis, we can identify whether bacterial contaminants are present in our sample. For example, we might amplify a specific gene from the bacteria. If the PCR result shows the gene is present and the gel shows a specific band, we know the sample is contaminated with that bacterium. Gel works by using an electric current to move the DNA through the gel matrix. The DNA fragments move toward the positive electrode because DNA has a negative charge.
What moves the DNA and how?
The electric current moves the DNA fragments with smaller fragments traveling faster and farther throughout the gel, while larger fragments move more slowly. The size-based separation creates a series of bands that we can analyze. Oh, I'm
That was a gunshot.
Can you flip the chicken?
What are you doing?
Versus cock.
Yep.
What?
I mean, hot dog
That's what it's
versus
called?
chicken. It's called chicken versus hot dog.
All right.
That's how I do.
4 or 5. 5 tries. Well, it took me 3 to do the harder one, so. I'm just cooler and better.
So what you really need to do is both of them
Yeah,
at
do
the
it.
same time.
Yeah, do it.
Right?
Go. Oh, almost a goat. Almost a goat.
That's pretty close.
Is it gonna be in our video some way, somehow?
Eye contact with the wiener wife of it.
I'm gonna make eye contact with the camera.
What separates the DNA, and why doesn't each band migrate the same distance? Don't put that in.
Gel itself is the medium that separates the DNA fragments. It's made of a substance called agarose, which creates a sieve-like structure. Smaller DNA fragments can slip through more easily, so they travel farther. Larger fragments face more resistance and move slower, so they don't travel as far. Okay.
To sum up, PCR allows us to amplify DNA, and gel electrophoresis helps us to analyze the results. Together, these techniques are powerful tools in genetics, microbiology, and many other fields. Thanks for watching, and if you learned something new, don't forget to like and subscribe for more science content.